So good evening guys and welcome back to the AgriAddict ICAR JRF plant science online sessions. So with the ongoing plant breeding classes, today we will be dealing with heterosis breeding. Okay. So can anyone say what do you mean by the term heterosis? How do you define it in any simple language? Can anyone define what do you mean by heterosis? Or you can also take the example of an hybrid. How do you define an hybrid? Anyone? In simple terms, yeah, someone wrote it is superiority of the F1 over parents. Okay, right. See, as we will be thinking only about yield or something which is beneficial for what do you call human beings, right? We just think about the yield terms and we will say that okay, superiority of the F1 over the parents in terms of the yield. But in fact, heterosis is nothing but said to be not only the superiority, but it also includes inferiority. Okay. For example, if we say, let us consider an uric acid content. Hope you guys know about this. Which crop have this? Also known as bone. So there is a neurotoxin in one crop, right? What is the crop? Do anyone know about that crop? The crop which contains the neurotoxin BOAA. Yeah. Anyone? What is the crop which contains that neurotoxin? Yeah, Lathyrus. Right? Someone wrote Lathyrus. So, for example, if there are two Lathyrus parents, so the neurotoxic content in this parent is 0.5 and this parent is 1. If you cross them, okay, if you cross them and you have got a progeny where the neurotoxic content is 0.09. Let us consider this. So, will we say this as a beneficial or superior? What do you call harmful?
Beneficial.
Yeah, right. So, actually, even though heterosis was defined as a superiority of F1 over both the parents, but it also has the other impression also. Okay, so when it comes to terms like this, in case of your neurotoxic contents, etc. So it directly means that RF1 is superior over both the parents. Right? So what causes heterosis? What is the genetic basis of the heterosis? What is the main reason why heterosis is being shown in plants? And which plants show more and more heterosis? Either it is self-pollinated or cross-pollinated. Which plants it is being expressed? So the genetic basis of the heterosis is, hope you all know, it is dominance. Okay, and it is seen in both self-pollinated as well as cross-pollinated crops, right? And do you know what are the two different theories proposed to explain this, the term heterosis? The two different theories that were proposed were, one is your dominance theory or dominance hypothesis, we can say, and the next one is the overdominance hypothesis. And we'll also see how we can fix the heterosis once the heterosis is seen in the progeny. So how can we fix and forward it to next generations? Okay, and we'll also see how to estimate the heterosis. What are the different methods to estimate them? And the ultimate product of your heterosis breeding is your hybrid, right? So we have different types of hybrids, and we will see what are the different types of hybrids and the role of what do you call this full form of MS? The role of MS in heterosis breeding or hybrid production. What is this MS? Anyone? What do
Well,
MS?
sterility.
Yeah. So what is the role of the male sterility in the heterosis breeding? So we will be seeing all these um, concepts in today's class. Okay. So coming to the term heterosis. So as your understanding is that the superiority of the F1 over parents. Okay. So who coined the term or the word heterosis? Who gave it? The term heterosis was given by a scientist named G.H. Shull. Okay, right. So Shull gave the term heterosis. He was working on the crop. It is a cross-pollinated crop, and it is also an example of your anemophily. So can anyone name it? Which is the crop on which Shull was working? It was a cross-pollinated crop. It shows anemophily, and it also has a geitonogamy type of pollination. Anyone?
Maize.
Yeah. So Shell was working on the maize, and we gave the term heterosis. And do anyone know what do you mean by the term luxuriance? Even though this is not in the syllabus, but it is important to know. So what do you mean by the term luxuriance? Anyone? What do you mean by the term luxuriance? Or did you heard about the term luxuriance? See, luxuriance is also some people call it as your pseudo heterosis. Okay, pseudo heterosis, where it is been seen that increase in the vegetative mass. Okay, so just remember it is also known as your pseudo heterosis. And as we discussed earlier, the genetic basis of heterosis is your dominance, and there are two theories or hypotheses which explains how this heterosis actually happen, right? So one is your dominance hypothesis, next one is your over dominance hypothesis. So who proposed the dominance hypothesis? Do anyone have idea who proposes the dominance hypothesis? This dominance hypothesis, it was given by a scientist named Davenport. Okay, and the support was from two other scientists, Kipling and Healy. So please to remember this one. It was proposed by a scientist named Davenport. Okay, right. So based on what points that the dominance hypothesis was explained, the only point where the dominance hypothesis was explained is according to this hypothesis, whatever heterosis we see is superiority of the superiority of your dominant alleles. Okay, Davenport said that whatever the heterosis we see in the plant, it is due to the superiority of the dominant alleles, while these dominant alleles they suppress the recessive alleles, and he said that these recessive alleles are deleterious. Okay, so just to remember this point that according to the dominance hypothesis, Davenport said that heterosis is due to superiority of the dominant alleles, which suppresses these recessive alleles. Okay, so for your clarified understanding, we will take an example. So let us consider that this gene, it gives 10 units of value to the yield. Okay, and in the dominant condition, dominant homozygous condition, and this gene in the dominant recessive condition, sorry, homozygous recessive condition, it gives 5 units. Okay, and vice versa is the case in this. So it gives you 5 units and it gives you 10 units. So when we cross between these two parents, we will eventually get a progeny with this. So someone tell me, if dominance hypothesis is working, what will be the value? So in case of this, the value is 15, and then in case of this, the value is also 15. So in case of progeny, if the dominance hypothesis is working, so what will be the value of your progeny? 20, right? Right. Someone wrote it as 20, because here the dominant allele, it suppresses the recessive one. Same in case of the other gene, and they eventually yield it up to 20 units, right? So can you understand the superiority of the dominant alleles causes the heterosis? Means if dominant alleles are more, then heterosis will be more. So in other terms, we can say that heterosis is directly proportional to the number of dominant alleles, right? Number of dominant alleles. This was one of the most important question in this dominance hypothesis. So heterosis is due to the dominant alleles, or heterosis is directly proportional to the number of your dominant alleles, right? This was the most important point that has been explained in this hypothesis by a scientist named Davenport. So, and there were some few objections to this dominance hypothesis. Okay, so one important objection is that, see, if you consider, okay, what do you call this? If you have a parent with 15 units of value, and if you cross with the other 15 units of value, so you said that you eventually got 20 units, right? But when you self this crop, what will be the progeny? If you self the F1, what will be the progeny genotypes? Anyone? Hello? Guys, am I audible? Hello? Hello? Yeah. Okay. So please to respond. I thought the connection was lost. Right. So if you self this F1 progeny, you'll be getting three different types of genotypes, right? This is one, and then this is the second condition, and this is the other condition, right? So when people self date, they observed that a pure heterotic individual, which means this one, right? A pure heterotic individual, right? Is it right? A pure heterotic individual, okay? This was not eventually observed. Is it okay? Is it okay, guys? Okay, I'll just make you more simple understanding it. So let us consider. See, if there are two alleles to a gene, okay, and if one allele it imparts 5 units and the second allele also imports the 5 units, and this imparts 2.5 and this 2.5. So similarly here 2.5 and then 2.5 and then the B with your 5 and then with this. So with this explanation, what will be the value of your F1? Fifteen. So why your A it eventually gives your 5 here, and this will be 2.5, and this whatever it gives it will give your 5 again, and your B it will be giving how much? 2.5. So your 7.5 and 7.5 gives you 15. So even your parents are 15 and even your other parent is 15. So can you see the heterosis in this case? Can you see the heterosis here? No, right? Yeah, no. So why the heterosis is not observed? Can anyone tell why the heterosis? We did not see any heterosis here. So you need to understand this very carefully. So why the heterosis here it is not observed? So our progeny it has all the two dominant alleles, but even though the heterosis is not observed, what might be the reason?
Absence of complete dominance.
Yeah, so because in this what is the gene action that we saw here?
Codominance.
No, not codominance, man. You're almost right in the first point, but we did not see any codominance here. There is no taking and giving regarding to codominance. Whatever we saw
Linkage.
here, no. See, just by looking at the book, don't tell the answers which are written there. See here. This gene it gives 10, and when we divide it give 5, and this with 5 2.5 2.5. So what was it done here? 5 plus 5 plus 2.5 plus 2.5. So what is been done here? What happened here? If you understand this, then it will be very easy to get the further concept. So what happened here, right? Additive gene action is been prevalent, right? Is it okay? Yeah, someone said it is additive. So which means your additive gene action is being seen. So but we are discussing about which gene action here. What is the gene action that we are discussing here? So we are looking at the dominance. So if we consider dominance, so with the same example, can anyone construct what might be the correct way to express it? So using the dominance itself. So what is the correct way to tell it? Anyone? So with the same example, right? Dominance. If you consider the example of dominance. So hope you guys understood that this is the additive one that we saw. So for example, there is a genotype like this, which has been crossed with this. So the progeny it will be ultimately right, and this it has imparted 2 units. This 2, this only 1, this again 1. whereas in case of this it has imparted 1 unit, 2 units, 2 units. So what will be the whole value of your parents, the individual parent? It will be eventually 6, whereas in case of this it should be 6. Then what is the value of your F1? It will be
8.
yeah. So now you can see heterosis, right? So as
Yeah.
someone defined at the starting of class that superiority of the F1 over both the parents. So in case of this, our value 8 is superior to both the parents. So on which concept was here the heterosis is being seen? So you can just consider, okay, even here the capital A is present. So we will consider it as, if you divide it is 1 and again 1. So you can take here 1, and if you from this it will be again 0.5. So can we do this mechanism here, 1 plus 0.5? Anyone? So can we again do the additive gene action here? No, yeah, because heterosis is based on dominance, which means if you just have a single dominant allele, so you'll eventually get 2 units. Whereas in case of this, you'll again get 2 units. Okay, right? You'll again get 2 units. You'll again get 2 units, right? So this is the mechanism. When he selfed it, okay, and you can obtain this type of individuals. Now we will again obtain all the genes in heterozygous condition, and you can obtain all the other genes in the homozygous conditions. So which means you can ultimately get an, what can you get? You can ultimately get an true heterotic individual, right? But this was not able to be seen. So what might be the reason? So this was not being seen. So what is the reason for that? See your what do you call that, the true heterotic individual for all the dominant genes. Okay, a true heterotic individual which has all the dominant genes was not able to be recorded in your F2, right? So the main reason for this is linkage, right? So someone said this was an objection, right? And Jones was a scientist who said the reason for this not to be seen in F2 is your linkage, and he proposed the dominance of linked gene hypothesis, which means it is not possible for all the dominant genes, okay, to get into a single plant, because the dominant gene of the dominant capital A gene might be linked with your recessive gene of B, or with the recessive gene of C, or this dominant capital or your gene of D, it might be linked with the recessive gene of C, but with the dominant gene of A. Is it okay? Hello, right? Did you understand? Is it okay? Yes, sir. I won't reply from other guys also. If you did not get it, sir, once again, please. Okay. See, when these two individuals were crossed, your progeny will be heterozygous in all the conditions. And when you self it, you can see three different types of genotypes with all the dominant homozygous condition, dominant your recessive homozygous condition, and then all the genes being in the heterozygous condition. But this was not observed in your F2. Okay, the main reason is that linkage. Someone told it was linkage. Okay, so if you want me to explain more clearly how this linkage acts, it is just that the dominant allele of gene A is linked with the recessive allele of your gene B, which means wherever the dominant allele of A goes, B also goes, right? Is it okay? Did you understand this particular point, that linkage? Example, if your dominant allele of wherever the dominant allele of your A goes, so eventually the recessive allele of your B goes. So can this individual be possible, this type of genotype? The genotype with all the homozygous dominance in nature. Is it possible, guys? No, right? So that is the reason why this was not observed. Some claimed it as objection, and to this objection, Jones was the scientist who said that this is not possible because of your linkage, and he gave the concept of dominance of linked gene hypothesis. So is it okay now? Right? Is it okay? As many are okay, but that linkage is not seen in parents' rights, sir. See, for example, I told you just that your what do you call, the capital allele of gene A is linked with your gene recessive allele of your gene B. Consider that the capital that is your dominant allele of gene A is linked with the recessive condition of gene C. Then is it satisfied? Can you satisfy with this? Someone said that, but that linkage is not seen in the parents, right? Okay, you can just take the other gene. So dominant allele of your gene A is tightly linked with the recessive allele of gene C. Is it okay then? Right? Hope I'm able to make you understand that. So if the dominant allele of the gene A is tightly linked with the recessive allele of gene C, then you can't see this type of genotype in the F2. And so, as this was an objection, Jones was a scientist who explained it with the help of dominance of linked gene hypothesis. That is, the dominant genes are not always linked with your dominant genes itself, means the dominant allele itself. They might be linked with your recessive alleles also of the other genes. So that is the reason why you are not able to see a true heterotic individual in the F2. Is it okay, guys? Now, okay. And the second objection to this dominance hypothesis is, so if you self an heterotic individual, you need to get three heterotic individuals and one recessive homozygous, right? So if the same gene action has been prevailing here, you said that it acts as 8 units. So even it should act as 8, and even it should also act as 8, right? So which means you will get one such type of plant, two this, and one this. So you need to get it in the 3:1 ratio, right? Which means if you draw a curve, it should be skewed towards that is among your four parts. So among four parts, how many parts? 3/4 of the part should be skewed, and only one part should be very less. So this, as yesterday's class was not recorded properly, so we will continue with the yesterday's class. Okay, so we discussed about heterosis breeding, and we are recorded up to the dominance hypothesis. And regarding overdominance hypothesis, you know that it was independently proposed by Schall and other scientists New East in the year 1908. Right. So the name itself indicates that this hypothesis is based on the concept of your overdominance. Right. So if you want to see in the genetic terms, this says that if you have a plant with this genotype and if you cross it with a plant with this genotype, So ultimately the F1, that is your hybrid, will be heterozygous. But according to overdominance hypothesis, we said that the one which causes your heterosis is the heterozygosis, right? Which is nothing but said to be your heterozygosis, which means the heterozygote itself gives more and more heterosis. For example, if the value of your AA is 2 and small bb is 1, so entire it will be 3. In case of here it is 1, in case of here it is 2, so entire it will be again 3. But whenever the plant gets in the heterozygote condition, okay, so this heterozygote, it is more than your normal homozygous dominant condition. So it tends to give you 3 and it also tends to give you 3. So the overall value will be 6. So, and we can say it heterosis because your F1 is superior to both the parents, right? So in this case, the heterosis is directly proportional to your heterozygosis. That means the heterozygous nature itself is enough to produce the heterosis, right? And there were many objections to this overdominance hypothesis because it was not possible in all the cases, right? And I also told you that if we have alleles like this, so these which we call as the divergent alleles, okay, whereas in case of your divergent alleles, so a1, a2 and a3, a4, okay, or a1, a4. So if we take these three examples, we can easily say that this will perform more, that is, this will have more heterosis, because more the divergence, the heterosis increases. And heterosis in case of your overdominance hypothesis is directly proportional to heterozygosis. This is point number one, important. And second is that, so more the divergent alleles, more will be your heterosis. Okay, so if A5 is there, so A1, A5 will be more heterotic when compared to your A1, A3, right? So if the divergence increases, the heterosis it gradually gets increased. So this is about your overdominance hypothesis. And then coming to the fixation of heterosis, I said you that there are 4 different mechanisms. One is your vegetative propagation. So once if you are able to cross between two vegetatively propagated plants, and if you get F1, so with the help of the propagule you can continue it. For example, you have a potato plant, two potato plants, you have crossed them and you are able to get a hybrid. So with the help of the tuber, if you just cut the eye of the tuber and if you again plant it in the soil, then you will get the same plant with the same genotype, which means the heterosis is fixed forever, right? So if you again cut the eye, you can fix the heterosis forever. So that is one mechanism, and the second mechanism is your apomixis. So which means even in this case also the progeny is similar to your female parent, which is similar to your female parent. So apomictic plants, if you have two apomictic plants and if you cross between them, if you are able to successfully cross between them, so if you generate the F1 with the help of apomixis mechanism, you can fix the heterosis. And the third system, which is balanced lethal system, which has been not discussed till now even in your UG classes, but just remember, even with the help of this balanced lethal system, we can fix heterosis. I'll just give you a hint how we can fix the heterosis. So in case of balanced lethal system, homozygotes will not survive, okay? Homozygotes will not survive. So every time what the progeny that survives is your heterozygote. So this is what we require for heterosis to perform, right? So in every cross and in every progeny, you will get only the heterozygotic individuals, and hence this is one of the mechanism where you can fix the heterosis. And there is other mechanism where you can also fix the heterosis. It is your polyploidy. Even with the help of polyploidy, you can get more and more heterosis. Right? So just remember, vegetative propagation, apomixis, balanced lethal system. and other mechanism is your polyploidy, and these three are very important. Okay, so this is how you guys will fix the heterosis. And then we also saw estimation of heterosis. So okay, your F1 hybrid is heterotic, that is, it is performing more superior when compared to both the parents. So how do you estimate it? For the estimation of this heterosis, there are three different types. One is your average heterobeltiosis and then useful heterosis. So as I told you yesterday that average heterosis, it is also known as your mid-parent heterosis or relative heterosis, right? So for the estimation of heterosis, the only formula is F1 minus, just assume a blank here, divided by blank into 100. So in this blank, we will be filling different parameters in the three cases. Whereas in case of your average heterosis, it is F1 minus your mid-parental value. The name itself indicates it is the mid-parent heterosis divided by mid-parent into 100. So for example, if you have a parent which gives a value of 6 and 6 tons of yield, and the other parent which is also giving you 5 tons of yield. So if you cross between them and you got a progeny which gives 10 tons of yield. So whereas in case of this, so 10 minus mid parent, that is nothing but the mean of your parents. So 6 plus 5 divided by 2, it will be 5.5. So 5.5 again divided by 5.5 into 100. So this gives the value of your average or mid-parent or relative heterosis. So whereas in case of your heterobeltiosis, this is also known as your better parent heterosis or superior parent heterosis. So let us consider the same example. So if you cross a plant which gives 6 tons of yield and other with your 5 tons of yield, and you got a progeny with 10 tons of yield. So whereas in case of this, the better parent will be this one, right? Because it is giving 6 tons, which is more than your other parent. So 10 minus 6 divided by 6 into 100. So this will give you the heterobeltiosis. So when coming to your useful heterosis, and this has many names such as your economic heterosis, also said to be your commercial heterosis, etc. So whereas in case of this, you need not to have take parents for calculating. Whereas in this case, you need to have a leading hybrid, as somebody told yesterday, or you can get a commercial check which is available in your area which is performing better. Okay, so we need to compare the F1 with the value of this leading hybrid or commercial check or standard check, whatever you call. Okay, so for example, let us consider the standard check is giving a yield of 9 tons. So 10 minus 9 divided by 9 into 100. So this gives you a commercial or economic or useful heterosis. And I also told you that this useful heterosis, it is given by Meredith and Bridge in the crop cotton. Please do note it is given by Meredith and Bridge in crop cotton. Right. So this is the concept of the estimation of heterosis. And then coming to inbreeding. So somebody told that inbreeding is mating between closely related species, right? So this is the definition for your inbreeding. So there is another term which is known as your inbreeding depression, which means continuous inbreeding causes reduction in vigor and fitness, right? So continuous inbreeding causes reduction in vigor and fitness, and this condition is known as your inbreeding depression. So high inbreeding depression is observed in two crops. One is your carrot, and what is the other crop that we discussed yesterday?
Lucerne.
Yeah, lucerne. It is also known as
Medicago.
alfalfa, right? Medicago, yeah, Medicago, alfalfa, etc. So high inbreeding depression is seen in carrot as well as your alfalfa. And we also discussed that self-pollinated and cross-pollinated crops. So inbreeding depression is observed in cross-pollinated crops, right? So more inbreeding depression is observed in cross-pollinated crops. In case of self, they do not have any inbreeding depression because they are always under selfing. And one another important question is the closest form of inbreeding. What do you call that? The closest form of inbreeding. Form of inbreeding. Which is the closest form of inbreeding? Anyone? We did not discuss it yesterday, but I'll just tell you. Which is the closest form of inbreeding? I'll just give you options like seed mating, selfing, cross-pollination, or getinogamy. Which will you select? Which is the closest
Selfing.
form? Selfing is the closest form of your inbreeding, right? So please to remember this was asked many number of times in your JRF examination. Okay, and even this high inbreeding depression is seen in carrot and alfalfa. This was also the most repeated, and in more inbreeding depression is seen in cross-pollinated crops, carrot as well as your alfalfa. Okay, so this is the concept of your inbreeding. And while preparing my notes for JRF, I also have the definition, the equation how to calculate the inbreeding. So it was as follows: F1 minus the value of your F2 divided by F1 into 100. Okay, right. So for example, if you have an F1 value of 100 quintal and if you have an F2 value of 80 quintal, so can we say this is inbreeding? It is your inbreeding depression. In some sense, we can say, right, because eventually the vigor is decreasing and ultimately even the yield, right? And even for if your F3, if it even goes below 80, for example, let us consider a value of 60, and then for F4, it is even decreasing to 10. So which means with inbreeding, that is your selfing, vigor and then the fitness is gradually decreasing. So this is the condition of your inbreeding depression. Right? So this is the concept of inbreeding. And then coming to hybrids, so we discussed there are mainly two types of hybrids. One is your intraspecific and interspecific. So intraspecific, which means within species. For example, Oryza sativa with a subspecies named something X into Oryza sativa with a subspecies named Y. So in case of this, the species is same. So this is known as your intraspecific, which means within the species. And the other type is your interspecific, which means which is outside your species. Okay, so what might be your example? Anyone? So someone told yesterday that cotton, so Gossypium hirsutum into Gossypium. This might be one example, or you can also consider we call that wheat as an example of a Triticum turgidum into Triticum monococcum, whatever. Okay, so if the species changes, right, hirsutum into barbendens of the genus remains same, but the species changes. Even here, the Triticum, the species remains same, but your the genus remains same, but your species it changes. So if species changes, then and if a hybrid is formed, such hybrids are known as your interspecific hybrids. So whereas in case of your intraspecific hybrid, we again classified them into three types. One is your single cross hybrid, double cross hybrid, and what do you call this? TCH.
Top cross.
No, even
Three-way
yesterday
cross
someone
hybrid.
yeah,
Three-way cross.
so it is your three-way cross hybrid, right? So single cross, just you have two parents, and if you perform a cross between them, you will obtain a hybrid. This is known as your single cross hybrid. Whereas in case of your three-way cross hybrid, you have performed a cross between two parents and you have obtained a single cross hybrid. And if you cross this single cross hybrid with another parent, this ultimate hybrid, so let us consider it as your F1 dash. So this is known as your three-way cross hybrid. In case of double cross hybrid, it is A into B, you obtain a single cross hybrid named F1, and then C into D, you obtain another single cross hybrid named F1 dash. So if you perform a cross between two single cross hybrids, and then if you obtain a hybrid H, so this is known as your double cross hybrid. So there are some formulas to define how many hybrids can be formed with a given number of parents. So if n parents are given, right, in case of single cross hybrid, so how many single cross hybrids can be obtained with the n number of parents? Can anyone just tell me? With the n
N
number of
into n
parents,
minus 1.
yeah,
Divided by
it is
2.
n into n minus 1. So I just asked you how many single cross hybrids are possible. So this is correct answer. If I ask you how many single cross hybrids are formed, but you need to exclude the reciprocals, then you need to divide it by 2. Okay. So only single cross hybrids. This.
Okay, sir.
If I ask you to exclude the reciprocals, then you need to divide it by 2. So the formula for three-way cross hybrid will be n into n minus 1 divided by 2 into n minus 2, or you can simply say n into n minus 1 into n minus 2 divided by 2. Whereas in case of your double cross hybrid, it is n into n minus 1 into n minus 2 into n minus 3 divided by 8, right?
8.
Yeah. So these are different number of hybrids that can be obtained in different intraspecific classification of hybrids. So with the given n number of your parents, and we also discussed that H4 hybrid, which was developed by C.T. Patel in 1970, was a cross between Gujarat 67 into, what was the another parent? What is your male parent?
American Heterosis.
American Heterosis, right? And then we also had the red gram hybrid. What was the red gram hybrid name, which was developed at ICAR in 1991?
ICPH8.
ICPH8. So what was the parentage?
MS Prabhat ICPL 61.
So this is your parentage of your ICPH8. So this was developed by ICAR, right? So and other hybrid I told you, Pusa RH10. Which hybrid is this? It belongs to which crop?
Rice.
It belongs to Rhiza sativa rice, right? So you need to gather the information of the different hybrids that are being released by Government of India every year. You need to have the information because these might be your important bits in the JRF examination. But most asked bit is your Pusa RH10. Okay, Pusa RH10. So please to remember, this is the hybrid of your rice plant. Right, so these are the different types of hybrid. And we also told that, for example, if we perform, this is small a, you obtain A dash, and then if you cross between these two single cross hybrids, you will obtain a double cross hybrid. So let us consider this name as your X. So okay, I obtained a double cross hybrid named X. So if I want to predict the performance of these double cross hybrids, then I need to go for mean value of single crosses which should be non-parental in nature, right? So how can I say it here? A into C plus A into D plus B into C plus B into D. Then I should divide it by. So it is saying mean, as we are considering 4 different type of non-parental crosses. So I need to divide it by 4. Then with the help of this formula, I can predict the performance of the double cross hybrid, right? So who gave the concept of predicting the double cross hybrid
Jenkins.
performance?
Jenkins.
It was given by Jenkins. Okay. Jenkins was the scientist. Right. So this is what we discussed yesterday, right? About hybrid breeding. So this is the entire concept of your heterosis breeding. Okay, and then the role of your male sterility. So if you want to perform a cross between two parents, you need to have a female parent and you also need to have a male parent. So always it will be very difficult because in case of your self-pollinated crops, there will be self-pollination. So we will be not able to cross between two parents. Okay, you can perform the emasculation in the one parent and you can go for crossing, but it would be a very manual and labor consuming, right? You need to go to every plant, you need to emasculate the anther, and you need to ensure that whether the anther got emasculated or not, and then you need to perform the cross. So if you want to overcome this barrier, you can just have a genetic mechanism that is your male sterility. So just select your male sterile parent and cross it with your male fertile parent. So you'll be able to produce the hybrid, right? So how do you designate this male sterile parent? How do you designate this male sterile parent? Anyone? Yeah, someone wrote A line, right? So this is your A line. And if I say A line and B line, there is one common thing between these two things. What is that? These two are, what do you call them?
Isogenic lines. Isogenic lines.
Okay. So these two are being defined as your isogenic lines. So this is the entire thing that we discussed about heterosis breeding. Is it okay, guys? Right. So this is what we discussed yesterday, right? And if you have any doubts in the heterosis breeding, you can ask me. If not, we will continue. Any doubts? Okay, I assume that you guys don't have any doubts. If you have, please just ask here. Okay.